The next speaker is Pantelis Rompolas. He's at the University of Pennsylvania, trained at Yale with Valentina Greco. And if you like movies, I think you're going to like this talk. It's Mechanisms of Hair Follicle Stem Cell Fate by Live Imaging.
Thanks a lot, George. I'd like to thank the organizers for inviting me to be part of this conference, and thank the chairs for putting together a fantastic meeting. I have no conflicts of interest to declare. And so, since stem cells were identified in the hair follicle more than 2 decades ago by George Kosarelis, there's been an enormous amount of work done that it's not possible to fit in one slide and cite them all, characterizing the niche and the different cell populations that take part in the niche and fueling the hair regeneration process. The current challenge that we face is that hair regeneration is intrinsically a very dynamic process where cells take a lot of activities and they assume many different fates. And so the challenge is that conventional histological approaches cannot capture these dynamic processes. And so the direction we took to address this challenge is to adopt a live imaging approach. So the question we posed was rather simple. Can we visualize the entire process of hair regeneration in the live intact skin? And so for that, we put together a live imaging system where we can visualize stem cells and other cell populations in the intact skin of live mice. And I have to stress that, and in the data that I'm going to present from now on, all are being, they were acquired from imaging live anesthetized mice from the intact skin. And so this is an example of how the process works. We anesthetize the mice and put them under the microscope, and we take a series of optical sections. In this example here, we use an epithelial reporter that was made originally by Doina Thunberg. It is an H2B fusion, so we see the nuclei of all the epithelial cells. And as you can see, taking optical sections starting from the top of the skin all the way down to the dermis, capturing the entire volume of the hair follicle. And so by taking these stacks over regular intervals, we can put them together and now see a continuity of events. In this example here, I'm showing you a movie that is over an 18-hour-long movie. So in this particular experiment, the mouse was under the microscope for almost 24 hours. As you can see, we can now capture activities that were not possible to be captured before by conventional histological approaches. As you can see here, for example, we can see that cells are proliferating in particular areas in the hair follicle. And I should say that this is a hair follicle looking at the outer root sheath in advanced growth stages. So this would be an advanced anagen phase. And so we can see that there are particular areas where cells assume certain activities. There's cell proliferation here. We can see cells dividing in a certain orientation, and also cells migrating to fuel the growth of the elongating follicle. And so the first question we asked is really, can we use this approach to understand what is the behavior of stem cells during hair regeneration? How is the hair growth organized, and which cells are contributing to the regeneration process? And so we collected a lot of these movies at different stages during the hair regeneration process. In this example here, you see 2 hair follicles at early anagen phase, and we can see cells dividing. So we quantified all these activities, and this is a summary of the data, and we started to see patterns emerging. In this example here, you can see that the majority of the cell divisions that we capture are primarily occurring in the lower part of the hair follicle and in close proximity with the dermal papilla, and also that these cell divisions have a certain orientation. We capture a number of other cell behaviors which bring up interesting cell biological questions, but the summary is that the cell activity seems to be specially regulated within the niche. And so the question now we wanted to ask is, looking long-term, what is the fate of the cells during the entire hair regeneration process? So starting from telogen, we essentially have a very small group of multipotent stem cells all these now have to somehow make fate decisions as to how to self-renew, how to generate progenitors, and then generate the 7 different lineages of the fully grown hair follicle. So how does this happen? So we use the inducible Cree system to label individual stem cells in the telogen follicle and distinguish them among the general population. So here in green you see all the epithelial cells of the hair follicle, and in red one cell being labeled. And we can have a large sample of hair follicles that we can use in a single mouse to do the tracing. And so what we do in this case is we can identify the same follicle in different stages during the hair cycle and now trace directly the fate of individual cells. In this example here, you see a cell in the lower part of the hair germ, and during the anagen phase, this cell produces a lineage, which is, in this case, a fully differentiated lineage of the hair shaft. And now we can also determine what is the fate of that lineage at the end of the hair cycle. So we traced a number of these cells, and to cut the long story short, what we found is that the location of individual cells at the telogen hair follicle could predict their fate during the hair growth process. So we found that cells in the hair germ almost exclusively differentiated and generated the lineages of the inner root sheath and hair shaft. Cells in the lower bulge region divided frequently and generating lineage that was confined in the outer root sheath. So these cells did not differentiate. And cells in the upper bulge region remained largely quiescent, although in some occasions this cell divided a limited amount of time. So putting these data together, what we found is that cell activity, especially regulated within the niche, is that location of a cell within the niche can predict its contribution to regeneration. So the question now that emerges after seeing these results is how the niche is organized after each cycle. Because obviously the location of a cell plays a significant role, so how is the entire niche organized after each hair cycle? What is the fate of the cells after the cycle is complete? And what is the origin of the new hair germ? The origin of the new hair germ is of particular importance because these are the cells that are going to produce the hair fiber in the next cycle. So for that, we looked into catagen, and here you see a movie of hair follicles undergoing catagen. You can see cells undergoing apoptosis, like in this example here. You can see the behavior of the cells that are differentiated and part of the inner root sheath and hair shaft. What did we learn? So the first thing we learned is that during the catagen process, the differentiated lineages continue to be excluded from the skin. So at this stage, the matrix has collapsed. There's no more proliferation taking place. But during the catagen process, we can still trace the inner lineages and the hair shaft continuing to move upwards and out from the skin. And in these frames from a movie, you can see the traction between the cells in the outer root sheath and the cells in the inner and hair shaft. And the changing the relative position as the inner lineages continue to be excluded from the skin and the outer root sheath remains at a constant plane. Looking at the cells in the outer root sheath, these cells by and large die by apoptosis. Here you can see a single example, and looking at a lot of those movies, we began to quantify how this apoptosis process takes place, which cells are eliminated first, and we found that cells are being eliminated from top up. So our question now is, who survives this process? And that has implications with who is going to put together the niche for the next cycle. And because the time frame is so short, catagen only takes 3 days in the mouse, and because we lack specific reporters to mark the different cell types and also have a temporal control over the labeling of the cells, we generated a mouse that hosts a photoactivatable reporter to be able to label cells with high spatiotemporal resolution. So the experiment we wanted to do is label cells in different regions of the catagen follicle and then see which of these cells survive the process and make up the new niche after the end of the cycle. And this is the mouse. Here, the photoactivatable reporter, which in this case is a photoactivatable M-cherry, is fused to H2B. And so normally at the normal state, this reporter does not fluoresce, but scanning a region with an activatable wavelength, we can activate this reporter. The activation is irreversible, and so now it functions as a regular fluorescent M-cherry. And this is a summary of these experiments. We labeled different areas of the hair follicle and traced these hair follicles up to the telogen state. And what we found is that cells in the upper outer root sheath are the ones that survive the entire catagen and the ones that make up the new hair germ. And I should just point out that in this experiment here, when we activate the reporter, essentially we label the cells in the entire volume of the hair follicle. So we label the cells both in the outer root sheath and the inner layers. And in this example here, you can see the separation of the two lineages. You can see cells here that were part of the inner root sheath and the hair shaft being extruded. And we can see, based on our previous result, that the elimination of the cells through apoptosis were happening from the top up, and so these were the last cells to encounter the wave of apoptosis, and the cells that eventually survived and made up the hair germ for the next cycle. And so the summary of these results is that differentiated cells in the inner root sheath and hair shaft lineages continue to be extruded through the end of catagen, that undifferentiated outer root sheath are eliminated by apoptosis, and that cells from the upper outer root sheath are the ones that survive catagen and make up the new hair germ. The next question we're going to ask is what are the requirements for the process of hair regeneration? Whether the niche is required and elements of the niche, and whether the stem cells themselves are required for the process. And so essentially this question aims to addressing which processes are regulated by intrinsic stem cell factors and which are regulated by extrinsic microenvironment regulators. The first thing we did is look at the mesenchymal niche. So from the original experiments of Colin Jahoda 2 decades ago, we know that the dermal papilla is an essential group of cells regulating the process of hair regeneration, and then from work of Bruce Morgan, Michael Rendell, and others, it's quite clear the importance of the dermal papilla in the hair regeneration process. What we wanted to do is to address directly whether the dermal papilla is required for the process of hair regeneration. And so what we did is we eliminated specifically the dermal papilla using a laser ablation approach. So for this particular experiment, we're using a femtosecond laser to very specifically eliminate cells of the dermal papilla. In this mouse, we have two reporters, an H2B GFP to mark the epithelial cells, and the left one RFP to label the dermal fibroblasts and the dermal papilla. Here you see the dermal papilla clearly visible, and after ablation, this group of cells has been eliminated. And this is a very specific method to kill cells in vivo, and as you can see, the neighboring cells as well as superficial layers are not affected by the ablation process. So what we found is when we ablated the dermal papilla and then followed the same follicle 7 days later, the ones that did not have the mesenchymal niche were essentially stuck in telogen. They couldn't initiate a new hair cycle, while neighboring hair follicles that had intact dermal papilla could normally activate and initiate the anagen phase. Now, what was interesting is that when we did the same experiment in catagen, we found that the dermal papilla and the mesenchymal niche also regulates the process of catagen. So when we eliminate the dermal papilla at the early stages of catagen, then the normal apoptosis of the outer root sheath does not take place. And we showed that this is mediated by TGF-2 signal, TGF-beta signal. So when we asked about what is the requirement of the niche, we found that the mesenchymal niche is indeed required for hair regeneration. How about the stem cells? When we did the same experiment, but now eliminating the cells in the balls, what we found is that the niche was able to reconstitute really quickly, and hair regeneration could proceed normally. So it was known from previous experiments of Miyumi Ito in George Cosarelli's lab that if you pluck a hair, you induce damage in the balls, and cells from the hair germ can reconstitute the balls in this case. But what we wanted to know is if there was a possibility that other cells can contribute to the reconstitution of the niche after the stem cells in the balls were ablated. And so in this experiment, we user-reported to label cells exclusively outside of the niche. Here you see a hair follicle where there are no labeled cells within the balls or the hair germ, but there are labeled cells in superficial layers, including the isthmus infundibulum, sebaceous gland, and also the interfollicular epidermis, which is not visible in this because the plane is at the dermal. So when we ablate the balls in this case, we found that there's a significant influx of cells from these superficial layers into the hair follicle. And not only that, when this hair follicle went into anagen and full growth, these cells participated in all the lineages of the mature hair follicle. So what we learned in this process is that stem cells of the niche can be dispensable because other cells outside of the niche can enter and acquire their fate. So to summarize everything that I showed you, what we found using live imaging to trace the processes that take place during the hair regeneration cycle, we found that the niche during the telogen phase has a compartmentalized organization, and that the location of individual cells within the niche could, by and large, predict what they were going to do during the process of regeneration. Cells in the hair germ committed differentiation and made all the lineages that are required for the mature hair fiber. Cells in the lower balls amplified and generated the lineage of the outer root sheath. And then when the growth process switched to the regression in catagen, what we saw is that the inner layers continue to be extruded from the skin. So these cells are essentially eliminated by terminal differentiation. Cells in the outer root sheath are eliminated by apoptosis, and these processes together form the niche for the next cycle. Now, there are a number of questions that remain to be addressed. There's a very high level of heterogeneity in the telogen hair follicle, which cannot be resolved with existing cell markers. So, new high-throughput genomic approaches and single-cell profiling is needed to characterize the heterogeneity of these cells. Also, a question is that there is an inherent hierarchy based on this that spans over multiple cycles. And one question is, how do these quiescent stem cells in the upper balls move down the hierarchy to contribute to regeneration? We heard from Doina's talk some of the possible mechanisms that can regulate this process. We also saw, by testing the different requirements in the regeneration process, that the dermal papilla is absolutely required for inducing hair growth. And we've heard wonderful talks yesterday from Bruce Morgan, and we're going to hear from Michael as well as to what is the nature of the crosstalk between the two populations. And obviously there's more to learn about that. Another interesting question is, in the experiment that we did, that we tested the requirement of the hair follicle stem cells, and we found that there is contribution from cells outside the niche. The question is, which are these cells that can enter the hair follicle niche and contribute to the hair growth? We don't know the identity and we don't know the mechanism of entry into the niche. So this is another interesting question. And also, whether these cells can fully replace those lost stem cells and be able to regenerate the hair throughout the life of the animal. So with that, I'd like to say that this entire work was done during my training in Valentina Greco's lab at Yale University. So I would like to especially thank her for being a fantastic mentor, and also thank my colleagues that contributed to this work, members of our lab, and also my funding during this process. And with that, I'd like to thank you. I'll be happy to take any questions.
Time for a few questions.
George? Pantelis, this is such a great technology and so many really fascinating insights. I'm curious about the DP killing experiments. Can you conclude whether the DP is initiating the catagen process, or can you only conclude that it's propagating at this stage? And the second question is with the killing during the regeneration phase, do you also kill the dermal sheath cells that you cannot dissociate the differential roles?
Right. Both great questions. So the first question is if the dermal papilla is required for initiating catagen, and we don't know that. The effect that we see is that when the DP is eliminated, the apoptosis process of the outer sheath is cannot proceed. So this is what we can conclude right now. For the next question is whether when we ablate the DP during the telogen phase, whether we are affecting dermal sheath cells as well. So this method of eliminating cells is not specific, genetically specific, meaning that we cannot differentiate cells that are in very close proximity but are of different lineages, dermal papilla cells from dermal sheath cells. So there's a possibility that we are also eliminating some of these cells as well. And with the existing reporters, unless we can fully differentiate a dermal sheath from dermal papilla cells, well, we cannot conclusively answer this question.
Mic 2, Valerie.
That was beautiful. So I'm curious about whether when you do the single cell labeling, whether the cells in the different domains stay within the domains. And for instance, do you ever see cells that migrate upwards towards the sebaceous gland?
So you mean when we label genetically or when we label it with a photoactivatable?
The photoactivatable would probably be the cleanest way, yeah.
If we see cells leaving the bulge and going into the sebaceous gland, we have not.
Bruce, mic 3.
So those photoactivatable lineage studies are just absolutely gorgeous. And I had mentioned in my talk that the matrix progenitors that we labeled, we could see them moving along the plane of the DP up into the higher rates. And that was technically limited because we label the bottom cells. We can only see them go up. So when you did these experiments, can you tell if that's a unidirectional property or whether they can actually go down as well?
Let me understand the question again. If you're saying when we label the cells with a photoactivatable reporter, yes.
So if you look at the cells that are actually touching the DP in the matrix, right? The ones at the bottom make the inner root sheath. And we label those with sonic RT2, and then we could see later in the hair cycle that they'd be up higher and they'd be making, for instance, the hair shaft.
Right.
But we couldn't tell if they could go down because we could only label the bottom ones. So now that you're sort of randomly labeling them,
right.
Can you tell whether it's
right? Yes.
directional?
Yes. Now I got the question. Except for the cells in the outer root sheath, we do not see cells going down.
Thanks.
Mic 2, Valerie.
Again, I must congratulate you on fantastic work. It's so nice to see the cells. My question was about when you were regenerating the mesenchyme area. Do you think those cells, I'm not so familiar with mice, but were those cells coming from outside of the sebaceous glands? Was it the mesenchyme or the dermal area from the spacious glands? You had two little round niches on the side of your follicle just above the bulge area.
So when we ablate the dermal papilla at the diligent stage, we never see the niche being reconstituted. So dermal papilla cells do not form, or dermal sheath cells, wherever these are coming from, do not form a new dermal papilla when the ablation takes place in diligent. When we ablate the niche in other stages, including catagen, we do see a reconstitution of the niche. And these most likely are coming from dermal sheath cells. So as Bruce stated yesterday, there is some temporal component where things can happen, but at particular stages during the hair cycle.
Angela, like 3.
Just beautiful. Thank you for the movies. So you've stared at probably thousands of movies, but just the few times I've seen the talk from you and Valentina, I've always wanted to ask, but it almost looks like, I mean, we draw the cells as if they're migrating straight up and straight down, but your movie gives the impression that they're spiraling. Is that
Right.
just an illusion? And if it's not an illusion, is there a directionality? Do they spiral counterclockwise on the way down and on the way up,
Yes.
or is it just my imagination?
So this is a fascinating question. There is a huge difference between the behavior of cells in the outer sheath and the clonality and how these clones expand versus the cells in the inner lineages that are differentiated. So the outer sheath, what we saw is that the clones tend to detach, and so these cells are generated in areas underneath the bulge and in a band, which we call the proliferative zone, that continues to go down as the hair follicle grows, but remains at a distance from the dermal papilla. And cells that are generated there by divisions that are happening perpendicular to the long axis of the growth are then now becoming mobile and migrating towards the end of the hair follicle. If we do the trace individual clones and do three-dimensional reconstruction, we often see these leaving a single plane. So it's what you described, maybe going around the hair follicle, but it's not a clear circular motion. It seems that it's almost chaotic. So they all migrate down, but they obviously do it individual from each other because we often see cells crossing paths. So it's not a clear stream going down, and this has implications of what are the connections between these cells, the lateral connections. There has to be certain fluidity to support these, but we don't understand the cellular mechanism. That's fascinating.
Great, thanks very much.